Thank you, Christian, for the short introduction. So my name is Carla Perez, and I am a PhD student at Umea University in Sweden. And as Christian said, I'm gonna talk about the co-hydrothermal carbonization of microalgae and digested sewage sludge. So microalgae cultivation using wastewater as growth media represents an effective way to remove nutrients, presenting also an efficient approach for biomass production than its cultivation in fresh water. However, an important factor to consider for reaching a sustainable and economically feasible use is to maximize the use of the residues generated. And since, like in this example, microalgae and sewage sludge, and since these both residues contain elevated moisture content, the hydrothermal carbonization was chosen here as a thermochemical treatment, as it might facilitate the efficient use of both feedstock within the same service system. I would like to make clear that the choice of these two residues is because they are available at a local wastewater treatment plant here in Umea, where I'm doing the PhD. So that's what we did here. We combined these both residues at the low, medium, and high carbonization temperature with different mixtures of the two materials, from just 0 to 100% of the sludge. So as we know, hydrochar is the biggest fraction formed during HTC, which is also the object of this study. So for that, it was of great importance for us to understand how the hydrochar is formed. So it's formed via two main pathways. First is the solid-solid conversion, in which the hydrochar maintains the original structure and morphology of the original feed. And then the soluble products transferred to the liquid phase due to hydrolysis, dehydration, and depolymerization would integrate some of those soluble products into the solid phase in the form of secondary char, which is extractable via condensation and repolymerization, and then promote the formation of heterocyclic structures in the main structure of the whole hydrochar. So based on this, what we aimed was to investigate the influence of microalgae and sludge blends on HTC performance at different temperatures, determine the composition, the properties of the hydrochars, and determine how much the composition of the secondary char. So all of this serves to explore the best exploitation alternatives of these materials. The most known, as we might know, is the solid fuel as a fertilizer or adsorption, for example. So before I start discussing, I would like to mention that since this study is still ongoing and we are analyzing data, I have hidden numbers. So what I'm gonna present is the most relevant aspects and trends that we have found. So in this first figure, we have the primary char recoveries, and we see that increasing the amount of sludge results in higher primary char recoveries, and thus a lower degradation and carbonization rate of the solid as the ratio of sludge increases, indicating that the organic fraction of a sludge is less reactive than microalgae under the same carbonization conditions. As a consequence of this, the amount of HTC liquor decreased gradually with the amount of the sludge, which gets more pronounced with the higher temperature also. Then we found that the extractive content, the secondary char, exhibits a change of trend with increasing temperature. So for example, at 180, the formation of extractives is inhibited by the addition of a sludge, which might be the direct consequence of the lower degradation rate of the solid and the resulting lower generation of soluble compounds that could lead to the formation of secondary char. So then as the carbonization temperature increases, the extractive content increases a little bit at 215, but then at 250, the condensation of soluble products from the liquid to the solid phase is favored by the presence of a sludge, which is in agreement with the higher decreasing rate of the HTC liquor at that temperature. So at 250 Celsius degrees, sewage sludge generates more soluble products that participate in condensation reactions and get incorporated in the solid product in comparison to microalgae. Then here, looking at more in detail the compositional differences of the hydrochars. The volatile matter in microalgae and sludge, it decreases both for both materials after HTC due to the commonly known and observed solubilization and volatilization of compounds to the liquid and gas phases with the increase of fixed carbon. However, it is clearly observed that microalgae reached a higher carbonization and aromatization with increasing temperature compared to the sludge. Here in the Van Crevelen diagram, we see that dehydration is the main carbonization reaction mechanism altering the carbon, hydrogen, and oxygen composition during HTC, which becomes more intense when the temperature rises. But dehydration is associated to the degradation of carbohydrates via the elimination of hydroxyl groups, and thus the carbohydrates may be the main fraction leading the changes that occurs during this Co-HTC system. We can also see that, again, microalgae reaches a greater carbonization extent than a sludge, supporting that a sludge is less reactive than microalgae under the same carbonization conditions. But also it's important to have clear that or to take into account that the carbohydrates content in a sludge are much lower compared to the amount in microalgae, which might be the reason for the lower reactivity of a sludge in this case. Here we can see the nitrogen recoveries, and for 180, the nitrogen recoveries decreased as a result of the inorganic ammonium salts and the hydrolysis of proteins and amino acids that were transferred to the liquid phase. So at this temperature, the solubilization of nitrogen compounds with increasing amount of a sludge is due to a higher content of nitrogen species that are more susceptible to be solubilized and hydrolyzed in a sludge compared to microalgae. Then increasing the HTC temperature there is a bit of an increase, which might be, especially when higher amounts of sludge were added, which might be the result of the extended hydrolysis of proteins to liquid products that are favored by temperature, and the deposition of nitrogen heterocyclic compounds as secondary char through the Maillard reaction. So the increase in nitrogen recovery at 250 might be aligned with the higher extractive formation rate observed at the beginning a few slides ago. Yeah. So then we conducted the thermogravimetric analysis on the hydrochars to understand and study the compositional, the fractions that change before and after HTC. So we can see clearly, we can see that the microalgae and sludge volatilization occurred in 3 and 4 stages respectively. This peak, I will talk about that at the end. So these peaks are associated to volatile matter fractions, and they seem to have similar thermal behavior. However, the weight loss in microalgae and reaction rate in microalgae is much bigger compared to sludge, especially because microalgae had more volatile matter fraction compared to a sludge. So if we go now peak by peak, the peak number 1 below 250 Celsius degrees, this one corresponds to the release of weakly bonded water molecules, along with the desorption of highly volatile substances, and also the alteration of lipid structures, the protein thermal unfolding, and incipient carbohydrate decomposition also occurred in this region. Then between 250 and 500, this region corresponds to the decomposition of biodegradable materials and organic compounds, again, proteins, carbohydrates, and lipids, but might include also the desorption of high molecular weight substances. So we see that microalgae exhibit 3 weight loss peaks associated to changes in proteins, but sewage sludge exhibit an additional peak not found in microalgae in the same temperature range where the decomposition of proteins happened. So this might be related to a different chemical nature of nitrogen species in sewage sludge that are not present in microalgae. For example, sewage sludge might contain nitrogen species in the form of macromolecular organic nitrogen that comes from proteins containing microorganisms and debris. More in detail, the HTC of microalgae resulted in the reduction of volatile matter. In this peak, we see that there is a reduction of the volatile matter after HTC, but then it results in the increase in a volatile matter in a mass gain, in a mass gain here of volatile matter. So these changes align well with the decomposition of the solid into the liquid and gas products, but also with the formation and deposition of high weight extractives onto the carbon solid matrix, and the integration of heterocyclic structures in the hydrochar. All these processes favored by temperature, carbonization temperature. If we look into the DTG profiles of the sludge, then the sewage sludge, the weight loss peaks 2, 3, and 4 gradually decrease after HTC, indicating the almost complete solubilization degradation of sludge under these HTC conditions. And conversely, this peak number 2 still appears after HTC of microalgae at the 3 temperatures, supporting that the sludge contain additional nitrogen species that are easier to hydrolyze. But HTC of a sludge at 215 and 250 gain a little bit of volatile matter in this peak. And this one is associated with the formation and deposition of high weight extractives onto the solid, as well as the integration of heterocyclic structures in the same matrix as I mentioned for microalgae. So in addition to this, HTC of a sludge at 250 Celsius degrees results in the formation of non-extractable volatile matter at 600 Celsius degrees, which is a peak that is not seen in microalgae, as it is just characteristic for hydrochars, sewage sludge hydrochars produced at higher temperatures than 200. This weight loss peak might be associated to complex nitrogen heterocyclic structures resulting from the Maillard reaction of nitrogen species contained in the sludge and their partial integration to the linked carbon network. Yeah, so here we have the DTG profiles of the carbonized hydrochar, the pristine, and the extracted hydrochar, which is the primary char once we have removed the secondary char. So the difference, for example, between the microalgae at 250 and the extracted one, this difference is the secondary char that we have extracted. And we see that the extractable volatile matter that is degraded in this stage is mostly associated to microalgae, as it decreases with higher amounts of sludge. So this indicates that microalgae gives higher amount of extractable low molecular weight volatile matter than sewage sludge. And then if we look at this area, this stage, we see that there is a little bit of a difference, a shift, as the extractives amount of high molecular weight are most associated to the presence of a sludge, as there is no change between the pristine and the extracted microalgae. So this will make more sense in the next slide. So here we have now the composition of the secondary char that was extracted and analyzed. And we found that it was composed to a higher extent with carboxylic acids, cholests, and ketones. So as we see in the figures, increasing amount of sludge significantly reduced the amount of carboxylic acids and ketones, with the remarkably notable increased content of cholests, which these ones exhibit the high molecular weight among the organic compounds identified, and the carboxylic acids that are more associated to microalgae, those represented the lower molecular weight compounds. So we can conclude that the fatty acids were the main precursors of secondary char, with the carboxylic acids and choles representing more than half of the total organic matter that we found. Then regarding nitrogen compounds, there was no increase at all in nitrogen compounds during HTC of a sludge. So all the assumptions that we contemplated until now could not be confirmed with this analysis. But we think that based on other results and what we found in the DTG is that the sludge led to nitrogen compounds that, after precipitated onto the solid matrix, were further carbonized and integrated into the primary char structure, contributing to this peak that it was present both in the pristine and extracted hydrochar, but also in other analyses that are not shown here because we need to further process the data. Yeah. Lastly, the addition of a sludge to microalgae had an impact on the composition of the secondary char and ultimately its toxic potential based on the organic compounds that we found. So hydrochar's toxicity, as we see in the figure, decreased with the higher amounts of when higher amounts of a sludge were added. And this is a matter of the lower amount of carboxylic acids formed due to the lower amounts of fatty acids originally present in the sludge. So these results are of high interest in the hypothetical case that these hydrochars were going to be used for environmental applications, for example. But as I said, I don't know if I said it, but this is just a preliminary observation, since more in-depth toxicity tests are required to really determine the toxicity of these materials. But it kind of gives us an indication of the type of materials that we have. So based on the linearity of the results that we pretty much got in all the analysis, we concluded that the Co-HTC of microalgae and the sludge is additive, as there was little synergy found between both materials during HTC. The secondary char formation increases with temperature and amount of a sludge. And we suspect, we cannot really confirm this yet, that there is some further carbonization of this nitrogen species in the sludge that become part of the main non-extractable hydrochar, the primary char. Based on the composition of the secondary char, it seems that sewage sludge hydrochars are more prone to be used in fertilizer applications, for example. However, there are more considerations that will need to be taken into account. And as I said, we are ongoing on this study. We need to understand more in depth the composition differences between the primary and secondary char to really exploit best the application alternatives of these hydrochar materials. So that's it. And since I have a bit of time, I would just like to mention and acknowledge all the people that it's involved in this project. especially my colleague Veronica Benavente, since we did both all the experiments together. So thank you for listening.
Thank you, Carla, for your presentation. Very interesting, and always nice to hear something about hydrothermal carbonization. And Albert had the first question that is a good start into the question and answer session. Can you describe the equipment used in the process a bit more? Because I think not everyone is really familiar with the hydrothermal carbonization equipment. So I guess he means the reactor.
Yeah, yeah, we use a closed pressurized reactor. I don't know exactly what you want to know. It's a wet technology, so we introduced the material wet, or we added more water to favor the carbonization reactor. So that's something that it's an advantage with HTC, that you don't have to dry the material. And in order to, the temperatures considered within the HTC range are up to 280, I would say, no more. Then you will go to mild pyrolysis, if I'm correct. I don't know exactly what you want to know about the.
So I think if that didn't answer the question, then Albert will write a follow-up in the chat, because I'm also not sure which part of the equipment he meant. Another question was from Juan about the toxicity tests that you run. Could you explain what kind of toxicity, or how did you measure toxicity, or toxicity to what?
As I said, we didn't run any toxicity tests. We just based the potential toxicity based on the organic composition that we found. So based on literature, for example, I actually wrote it here. The potential toxic compounds are phenolic and organic acids, ketones and furans, and benzene derivatives. So some of those compounds have been shown to be microbial and seed germination inhibitors and toxic to plants. So it's just what I have shown is just a preliminary toxic potential. We of course should do more toxicity tests, which in fact they were, at least for microalgae, they were going to be done in Edinburgh.
Oh, okay, good. Good
Yes.
to know that they will be done here. And I had a question to the DTG curve. So you did it in nitrogen atmosphere.
Yes.
And did you look at the ash-free? Did you calculate the ash-free curves? So subtracting the ash from, or maybe you did that already, or not?
The ash-free in the DTG.
Yeah, so that you subtracted the ash from the percentage calculation. Because I was wondering for the sewage sludge, how that would look if the percentage is on an ash-free basis. I'm not sure. That was
I actually am not sure. I can actually look later and tell you, because as I said, this was done with my colleague Veronica, and she was doing this, so I can